I'd now like to go over the more modern technique of DNA sequencing where you're able to use just a single reaction and you are not required to use radioactive labeling for this. And so this makes it a much more attractive, streamlined method of DNA sequencing. Now, having said that, it's also to be recognized that even today, in 2010, there are more advanced techniques that have been developed and are used, but this is a pretty standard technique, and when we do our DNA sequencing in lab, we will actually obtain data that was done via a method very similar to what I'm going to describe here. So to start this discussion, basically look in this portion of the slide where in your reaction mixture you put your template of unknown sequence and you put a primer. Now, how do you get a primer if you don't know the sequence? Well, typically the template has some region of sequence that you know and are able to generate a primer for, and that the primer will then anneal to the template just like in our previous reaction. Now, the difference here is that when we add in our enzyme and our DNTPs and then our dideoxy-NTPs, we add them all together in one reaction. Okay, now how are we able to do that? Well, it turns out that these DD-NTPs, or the dideoxys, each have a fluorescent label, and the different bases have different fluorescent labels. So the A base has something that fluoresces at one wavelength, the T base, and then here we're talking about the dideoxy-Ts. The dideoxy-Ts have a wavelength that fluoresces at a different wavelength, and then so forth. Now, what that allows you to do is it allows you to visualize fragments that end in A at a particular wavelength. And so when you carry out your DNA synthesis with this mixture, you will get a set of fragments. And these fragments you are able to separate, and the way you separate them is similar to the way we separate the other, except instead of having a big flat gel, what you're going to use is a really tiny little gel that you place inside of a tube that's called a capillary tube, but it's a very long capillary tube. And then this gel, this polyacrylamide gel, is inside a long capillary tube, and then you add your sample to the top. Your sample runs down the gel, and as it runs down the gel, it gets separated into individual bands. And the individual bands are then detected via the fluorescence that they emit. So you excite them with a laser, and then they will emit a particular wavelength fluorescent. And so when you're looking at an A, so you have basically 4 channels. You look at these 4 different wavelengths. Well, whenever you see an intensity at a wavelength that corresponds to A, you'll get a peak at A at that wavelength. So there's all the red peaks are recorded on one channel, all the blue peaks are recorded on another channel, and so forth. Then you take all of those different channels, the 4 different channels, you combine them together, and you get a sequence, and you know what base goes where in the sequence based on its particular color. So you can see the sequence here. We've got a blue peak, a red peak, a yellow peak. So that's CCTG.